Hello friends, welcome to BMH Learning. In this video, I am going to discuss about antimicrobial properties of plant extract. Introduction. Microorganisms are the biggest enemy when the concern is about food safety. These minute creatures spoil all types of food and cause loss and wastage of food even in the developed countries. If we make an estimation, it comes to the fact that globally, approximately every year, food losses and waste reach up to 40% due to various factors, and that includes the spoilage by these tiny living things. I mean the microorganisms. The common types of microorganisms that cause the spoilage of food products are bacteria, yeast, and molds. Apart from food spoilage, the microorganisms are also responsible for foodborne diseases. So these diseases are caused by consumption of contaminated food products. So we need to save our food from these enemies. Now, the traditional method to prevent food spoilage and their etiological agent is actually the use of chemical preservatives. But there are a lot of problems with this method. The continuous use of these chemicals led to the accumulation of chemical residues of these preservatives in food and feed chain. It has resulted in development of microbial resistance to these chemicals. Apart from these, there are a number of side effects on human health. So we should find another way to save our food and hence our health. So, due to these concerns, efforts have been made to develop food preservatives which are potentially effective, healthy, safer, and most importantly, they are natural. So this is in context of use of plant extracts as antimicrobial agents for food preservation. Traditionally, crude extracts from different parts of medicinal plants were used for treatments of various human diseases. These medicinal plants actually have various phytochemicals, mainly flavonoids, alkaloids, tannins, and terpenoids. And because of these phytochemicals, the plant possesses antimicrobial and antioxidant properties. There are some plant species whose antimicrobial activities have been widely researched. For example, we have cinnamon, garlic, basil, curry, ginger, sage, mustard, etc. Now, these plant species prevent the growth of a wide range of gram-positive and gram-negative bacteria. Specifically saying, the growth of Candida albicans is effectively inhibited by use of alcoholic extract of curry leaves. The thiamyl extract can reduce the growth of Candida albicans and also Pseudomonas aeruginosa. You can find a number of such examples in scientific literature. Now let's move to the experimental part. First thing we have to do is to prepare plant extract. As in this video, I am showing you the general procedure so we can choose any medicinal plant which possesses antimicrobial properties. So, we need leaves of this medicinal plant. In order to make the extract, we have to crush the leaves. So, for that we require pestle and mortar. Next we have to add 2 to 3 ml of ethanol to the crushed leaves. Then we have to mix it thoroughly. Alternatively, we can use column chromatography, in which we wrap the leaves in a filter paper. But remember, you have to dry the leaves before doing this. Then filter paper containing the leaves is placed in the chromatography column. In the next step, ethanol is added from the top of the column. As the ethanol comes down the column, it actually passes through the leaves, and hence the eluted ethanol has plant extract in it. The ethanol is allowed to evaporate and we get this, the pure extract. Next thing we have to do is to check the antimicrobial activity of the plant extract. So for that purpose, we are using disk diffuser method. So let's prepare the agar solution for this. Let the solution get cool at room temperature. The solution is having a specific type of bacteria, or you can say other microbe against which we have chosen the plant extract. As this is a general procedure, I am not specifying here the name of the bacteria and the plant. The molten agar with the bacteria is layered in a petri dish. The petri dish is then incubated at 25 degrees Celsius. It's because this is the ideal temperature for bacterial growth. After this, we need paper disc. You can either put the paper disc in the mixture made in the pestle and mortar, or you can dip it in the solution from chromatography column. In either way, we are getting some plant extract on the paper disc. The disc is then placed on the petri plate. Apart from this extract treated paper disc, we place one more paper disc as a control. It's used for the comparison. This paper disc is only soaked in alcohol. Finally, we leave it for 2 to 3 days and then compare the diameter of the inhibition zone which is formed around the paper disc. If the plant being used is having a good antimicrobial property, the diameter of the inhibition zone will be larger. This was all. Thanks for watching.